Hi all. So my topic of discussion is salvage pathway of pyrimidines. So the salvage pathway recycles free pyrimidines formed from the nucleic acid degradation. So salvage means to rescue. So it is the rescuing or recycling of free pyrimidines that are formed from the degradation of nucleic acids. So nucleic acids are DNA and or RNA. So the breakdown or degradation of DNA or RNA produces uh, nitrogen bases like thymine, cytosine or uracil. So breakdown of DNA produces thymine and cytosine, whereas breakdown of RNA produces cytosine and uracil. Okay, so now this is where the salvage pathway starts. So now we have three nitrogen bases: thymine, cytosine and uracil. So this first we take thymine. So this thymine gets transformed into deoxythymidine. Deoxythymidine. So it is by the enzyme thymidine phosphorylase. So in this reaction, thymine gets combined with the deoxyribose 1-phosphate. Thymine gets combined with deoxyribose 1-phosphate. Deoxyribose 1-phosphate. And it forms deoxythymidine. Okay, so thymine, which is a nitrogen base, it now became a nucleotide. So thymine, it got added a molecule of sugar and a phosphate group got added into the thymine nitrogen base, and it became deoxythymidine, which is a nucleotide. So this reaction is catalyzed by thymidine phosphorylase. So once again, thymine plus deoxyribose 1-phosphate in presence of thymidine phosphorylase gives us deoxythymidine. Now this deoxythymidine in the presence of or by the action of kinase enzyme, that is deoxythymidine kinase enzyme forms deoxy TMP, that is deoxythymidine monophosphate, deoxythymidine monophosphate, DTMP. So in this reaction, a molecule of ATP is utilized and ADP is formed as the byproduct. So deoxythymidine in presence of kinase enzyme forms DTMP, and a molecule of ATP is utilized and ADP is formed as the byproduct. So we now got DTMP, which is the required nucleotide for DNA synthesis, deoxythymidine monophosphate. Now we come to uracil. So thymine, so the nitrogen base thymine is coming from only DNA, and the nitrogen base uracil is coming only from the RNA. So now uracil, in the case of uracil, the enzyme is uridine phosphorylase. In thymine it was thymidine phosphorylase, and here it is uridine phosphorylase. Okay, and the sugar is ribose 1-phosphate. Because it is RNA, so there won't be any deoxy. So deoxy will not be there. So it will be ribose 1-phosphate. So uracil plus ribose 1-phosphate in presence of uridine phosphorylase becomes uridine. Okay, uracil plus ribose 1-phosphate in presence of uridine phosphorylase becomes uridine. Okay, now this uridine in presence of kinase becomes UMP, and a molecule of UTP becomes ADP in this. ATP becomes ADP in this reaction. It is similar to the previous reaction that we saw here. It is similar, no change, only the reactant and the product is different. All the intermediates and the intermediates in the sense ATP, ADP and kinase enzyme all are same. So I hope that is clear. And now coming into the nitrogen base cytosine. So this cytosine is produced from both the breakdown of DNA as well as RNA. So this cytosine can undergo deamination, and uracil can be produced. In the structure of cytosine and uracil, the only difference is cytosine has got an extra NH2 group, and uracil has got a double bond oxygen in that position. So that is the only difference. So if cytosine undergoes deamination and an NH2 group is removed, then we get uracil. Okay, so when we look into the formation of CMP, then the cytosine combines with PRPP. We have seen PRPP in lot of places. So PRPP means phosphoribosyl pyrophosphate. Phosphoribosyl pyrophosphate. So cytosine plus phosphoribosyl pyrophosphate or PRPP in presence of PRTAs. PRTA means phosphoribosyl transferase. Phosphoribosyl transferase. Okay, so in presence of phosphoribosyl transferase, it becomes CMP. So cytosine in presence of PRTase combines with PRPP to form CMP. Now we got CMP. So CMP gets converted into CDP and then it gets converted into CTP, and this is utilized in RNA synthesis. Okay, for DNA synthesis we required DCMP or DCTP, that is DCMP, which can be converted into DCTP. So this is required for DNA synthesis. So for that, this cytosine combines with deoxyribose 1-phosphate, similar to this reaction here. So cytosine plus deoxyribose 1-phosphate in the presence of the enzyme cytidine phosphorylase, similar to thymidine phosphorylase. Here we have cytidine phosphorylase. The cytosine forms deoxycytidine. Here it was deoxythymidine. Here it is deoxycytidine. So cytosine plus deoxyribose 1-phosphate in presence of cytidine phosphorylase gives deoxycytidine. This deoxycytidine in presence of kinase and ATP forms DCMP, similar to this reaction here. Okay, now these are all the monophosphates. For DNA and RNA formation or synthesis, we require triphosphates, TPs. So these monophosphates undergoes phosphorylation twice by the action of kinase enzyme and in the presence of ATP to form the respective triphosphates. Okay, so DTMP becomes DTTP, DTTP, CTP, UTP and DCTP. So DTMP becomes DTTP, CMP becomes CTP, UMP becomes UTP and DCMP becomes DCTP. So that is about the salvage pathway of pyrimidine. Now about the significance of the salvage pathway. It maintains cellular balance between pyrimidine nucleosides and nucleotides. Because here we can see this pathway contains, this reactions contains nucleotides as well as nucleosides as the intermediates. So this pathway helps in the maintenance of the cellular balance, helps in maintaining the cellular balance of pyrimidine nucleosides and pyrimidine nucleotides. So that is the significance of salvage pathway. So I hope that is clear. Thank you.